So what I'm going to talk to you about today is studying HIV through next generation or second generation DNA sequencing. For some of you who may have come to this class in the past, this is a topic that is near and dear to my heart and one that I've been talking about for the last several years. And it's really interesting to me when I put this talk together to see how the landscape has been changing from the first time I talked about sequencing 2 years ago to the talk that I'm going to tell you about today. I've also learned through giving these talks at this meeting before is that there's a wide diversity of viewpoints and occupations represented. And so instead of talking just about some of the basic science work that we've been doing with next generation sequencing, I'm going to break up my talk today into an introduction to DNA sequencing as of right now, and then I'm going to go into 3 short case studies that illustrate different aspects of how this technology can be used to study different types of HIV. So I'm going to talk about how clinical researchers could benefit from rapid, accurate, and economical HLA typing, how those of you studying epidemiology may benefit from being able to do drug resistance genotyping on entire communities of treatment-experienced persons. And then finally, I'll talk about some of the basic research that we've been using this technology for to understand how T-cell responses are or are not working against HIV infections. So to start with the summary, I know that this might be hard to read in the back of the room, but just focus on the boxes. Red means bad or not so good. Green means very good. And this is the sequencing standard as of about 2006, 2007. Everyone was using Sanger dideoxy sequencing, which had been the industry standard for going on about 20 years. We had made engineering improvements to the Sanger technology that allowed us to run machines that Sanger sequenced 24 hours a day, 7 days a week. The Broad Institute and other genome centers had warehouses full of Sanger sequencers that were generating data in an assembly line factory type approach. But the same underlying technology was used in about 2005, 2006, as had been used in 1985, 1986. In the last several years, there's been the introduction of new sequencing technologies that have been very disruptive. These technologies generate massive amounts of data relative to Sanger sequencing, and I've listed some of these technologies here and focused just on these 2 columns. Whereas a fully operational Sanger enterprise could generate about 6 megabases of sequence a day, some of these new technologies generate about 1,000 times as much data, 5,000 megabases a day, and this number is increasing exponentially. At the same time as the throughput has been increasing, the cost per base of acquiring the data has been plummeting. So here it was about, I think that's about 50 cents or $500 a megabase, and now it's less than 50 cents a megabase to acquire this data. So the marginal cost for acquiring sequencing data is dropping to near zero. And within a few years, the cost of sequencing data is going to be essentially zero. And the cost for generating sequence is going to be borne by the labor in obtaining the sample and preparing it for sequencing and analyzing the sequencing, not at all in the generating sequence itself. And this is going to be a very enabling technology, as I hope to show you. It's also a very fluid and dynamic marketplace. This Helicos machine, which I showed, is likely not going to ever be commercially viable. Helicos has been delisted from the American NASDAQ stock exchange. So just in the last couple of months since this, I abstracted the slide, one of the technologies has essentially disappeared. And so right now I think it's safe to say that the leaders are Illumina, Solid, and 454 Roche. And what I'm going to talk about today is principally work that we've been doing with the 454 Roche chemistry. Again, these technologies are characterized by massive throughput, and the ones that generate the highest throughput, Illumina and Solid, generate very short read lengths per individual read, generally less than about 200 base pairs per sequence. These are expensive instruments. They tend to be consolidated at genome centers and academic core labs, and they require a high degree of specialized expertise in terms of preparing the samples, generating the sequence, and then analyzing the data. And Shelby talked earlier about the project that we've been doing with Baylor's Genome Center to sequence 2 macaque genomes by Illumina. This is a brief timeline of the project. We started the project last March, and we're expecting the data this spring. And we will be getting in a massive amount of data that, as Shelby said, we don't really know how we're going to analyze it. You can see that doing this sort of project in coordination with a genome center is a lengthy process, though one that we think is well worthwhile in certain cases. What I'm going to talk about today, though, isn't the sort of sequencing that can be done at a genome center. What I'm going to talk about is the sort of sequencing that one can do in an individual academic laboratory using desktop instrumentation, which is to sequencing as the personal computer was to computing back in the late 1970s, 1980s. This is what I'm calling laboratory next-generation sequencing, and there's several platforms that fall into this category. The Roche 454 has an instrument called the GS Junior. Ion Torrent has their personal genome analyzer, and Pacific Biosciences has a machine that is a high-throughput machine that nonetheless has many of the attributes of laboratory sequencers, so I kept it in this category. These machines are characterized by dramatically reduced throughput. Instead of generating millions or billions of data points per experiment, you generate approximately 100,000 sequencing reads. But these read lengths are often longer than the read lengths that are generated by Illumina Solid, generally longer than 300 base pairs. And so what I'm going to talk about today is how we've used this technology to sequence large numbers of samples in single instrument runs on these desktop instruments. The instrumentation itself is less expensive than the sort of genome center instrumentation. The GS Junior, for example, costs about $100,000. But a trade-off for having this sort of technology in your lab is that there is more of a do-it-yourself ethos to using it. The laboratory staff are responsible for sample preparation, sequencing preparation, maintaining the instrument, acquiring the data, and then analyzing the data when it comes off. So here's a picture of the Roche 454 GS Junior. Almost all of the data that I'm going to talk about today was acquired on the GS Junior that we've had in our lab for the last year. And you can compare it to the Roche Flex machine, which is the genome center version. So this is the mainframe, and this is the PC. And the data we're going to talk about today came off the PC. You can see from this example workflow how having this sort of access in a laboratory can change the way that you think about sequencing projects. We can think about a project over the weekend, start it on Monday, and have our data by Friday. It's empowering to be able to generate this sort of data so quickly, assuming, of course, that you've already put in the energy to build up the infrastructure necessary to acquire this sort of data. So what's been our experience? We've run the machine about 168 times. As of the time I put together these metrics, we'd run it about 150 times. Most of these were runs with actual samples. We had acquired 9.5 million high-quality sequence reads, 3.5 billion base pairs of sequence data. This is a very impressive statistic. Anytime you're not talking about this at the Broad Institute, they tend to laugh at you when you show them this sort of data. In each run, we get between about 60 and 100,000 sequencing reads that are quite long by next-generation sequencing standards, 300 to 400 base pairs. And perhaps the most important statistic for those who are contemplating adapting this sort of technology in their work is that we've trained 25 different users on the use of this machine, including 4 undergraduates and 1 high school graduate. So the barrier and the learning curve is not nearly as steep as it is for using many of the other high-throughput sequencing instruments. Those 150 sequencing runs have supported a variety of projects, some of which are listed here. We've used it to understand immunogenetics in non-human primates. We've used it to look at MHC expression in distinct lymphocyte subsets. As Shelby briefly alluded to earlier, we've been looking at KIRs, and just this week we got our first T-cell receptor data off the machine. We've worked with people who study the ecology of wild primates and the discovery of novel viruses to discover new arterioviruses in red colobus monkeys. And we're also using this technology to understand the ecology of naturally occurring SIVs in wild primates, and we've been able to identify viral RNA from SIV in an African green monkey, a black and white colobus, and a red colobus. We've been able to study HIV and SIV evolution both within short regions, which allow us to look at things like drug resistance, which I'll talk about, and immune escape. And we've also generated protocols that allow us to sequence RNA viruses in their entirety. And we've used this to sequence HIV and SIV. The HIV work was done in collaboration with many of you in this room, and Esper, of course. In collaboration with Tom Friedrich, we've sequenced entire influenza genomes. And in collaboration, again, with many of you, we've been sequencing dengue virus genomes to assess intrahost and interhost evolution. So there's a wide variety of projects that can be supported on this sort of instrumentation. And so now I'd like to go into the case studies. The first one I'm going to talk about is the use of this technology for rapid, accurate, and economical human leukocyte antigen genotyping. I'm going to show this slide because Danny was talking about how much he liked it during the coffee break, and I agree with him. I think this is a great slide that really shows in individuals who are HIV controllers the importance of the HLA region in determining one's ability to spontaneously control viral replication. So it's very important to understand the HLA genotype of patients who are in research studies. And it's important to understand how HLA-mediated T-cell responses and potentially NK cell-related responses affiliated with the HLA drive evolution and impact pathogenesis. Now, one of the challenges of obtaining this sort of data was made obvious to me about 2 years ago. So this was a picture of my lab as of about August. And this is Dawn back in the back corner. Some of you have met her. A few of you who've been to Wisconsin have actually worked with her. And she studies HIV-positive patients in Madison and Brazil and Uganda. And in the front row, this is Simon. He helped us develop the MHC typing in macaque monkeys that preceded our study of HLA genetics in people. And finally, we have Bill. And Bill runs the molecular diagnostics lab at our hospital. When we asked to genotype some of the patients as in one of our HIV research studies, he told us it would be about $500 per patient to obtain 4-digit HLA class 1 typing. We had about 20 patients to genotype, and so we decided that instead of spending the money on getting the genotypes from these patients, we would just do it ourselves, or at least spend the money trying to do it ourselves, and that's what we did. As Shelby said earlier, the HLA class 1 locus is extraordinarily polymorphic. It's the most polymorphic genetic locus currently known, with more than 4,500 class 1 alleles. And the gold standard for determining which of these alleles are in an individual patient is to sequence them, is to go into each of the individual domains of the HLA molecules and sequence them one at a time. Historically, this has been done by Sanger sequencing, but this is very, very expensive. And so most hospitals, including ours, use single nucleotide polymorphism-based technologies for their HLA typing, which are less expensive, but are also less accurate, as I'll show you in a minute. We thought of adapting this to 454 because if we were able to eliminate the sequencing costs or dramatically reduce the sequencing costs, we might be able to generate the HLA types of patients much more economically than was possible in the past. So you can think of this entire project as we did it because we're cheap. And this just illustrates how, as you increase the number of patients who you study simultaneously in an instrument run, the cost of the sequencing per patient drops dramatically, such that if you have 192 or 576 patients, and this is about the scale that we're at now, the sequencing cost is measured in the tens of dollars. So several groups have now gone and have adapted that Sanger-based sequencing, where each exon is sequenced individually, to 454. This approach has the advantage of being somewhat more cost-effective, but it is difficult to pool and multiplex hundreds of amplicons together, as is necessary when you need to do 9 amplicons per patient. And again, we're cheap, so we wanted to see, could we try to do this using 1 amplicon per patient? And to do this, we developed a method that we termed cDNA sequence-based typing, where we used a single amplicon that utilizes conserved sites in the HLA class 1 open reading frames. And we let nature do the work for us. We let nature put the exons together. We sequence across the intron boundaries because they're simply not there. And then we can sequence all of the class 1 alleles simultaneously using pan class 1 priming. And we can take advantage of the fact that in the HLA class 1 genes, there are these regions of relatively good sequence conservation where all 4,500 known class 1 alleles have similar sequence motifs. This allows us to situate primers in these conserved regions, which are shown here with arrows. When we first started, we generated a 581 base pair amplicon that spans from this arrow to this arrow. And as you can see, there's an enormous amount of diversity in the amplicon that we're able to generate. And so to test the performance of this amplicon, what we did is we contacted the International Histocompatibility Working Group, and we got a set of about 40 HLA lines that are their reference cell lines that they use to validate all new technologies. And we asked how well this approach would capture the typing. We did all of this in a 1/16th of a single 454 plate. And you don't have to worry about what all the typings are. Just know that there's a lot of HLA types in here. Let's focus instead on the colors. Green here means that the typing that we got was concordant with the typing that was in the reference cell bank. And pink and yellow are typings that were discordant. So we were happy about the fact that 186 of the 192 alleles were concordant, but we were concerned that there were 6 discordances between our typing and the working group typing. So we contacted the working group, and we tried to resolve these differences. And what we actually discovered was that the 454 typing had accurately gotten the calls, but that the working group cell lines had been mistyped. And when they reconfirmed the typing themselves, it turned out that our typing was right. In 3 cases, the lineages had been mistyped by the other technologies that they had been using. We identified 4 lineage correct alleles where we were right to 2 digits. We had the same typing to 2 digits, but the 4 and 6 digit typing was different. It turned out there again that the 454 was right and the cell lines were wrong. And this has now been quite confidently confirmed by the IHWG. And so we can safely say that we were about 100% concordant, and we actually resolved errors in the IHWG typing. So we really like this method and believe that it can offer very cost-effective genotyping. Now, a limitation to this method is that this 581 base pair amplicon is not truly unambiguous. What that means is that we can get down to the 2-digit HLA type. Is someone B27, B35, with a high degree of accuracy, but we can't always resolve whether someone is B2701 or B2702, or HLA A201 or A202. In order to do that, we need more sequence coverage. We need to be able to study these sites of variability that are outside of our 500 base pair amplicon. And so to do this, we generated another amplicon that we call our 1KB amplicon. And we're now using a 2-stage approach to obtaining the data. We first sequence with this 580 base pair amplicon to give us the lineages, and then when needed, we layer on the additional data from this overlapping fragment that stretches into the 581 base pair amplicon. And so we're essentially covering nearly the entire open reading frame of the HLA when we're doing this genotyping. And we believe that as technology for sequencing improves and we get longer sequencing reads, we'll eventually be able to drop this amplicon and do our genotyping again using only a single amplicon that stretches approximately 1 kb. Right, so the question is, since the 454 reads are only 400 base pairs long, how do you sequence a 1 kb amplicon? The answer is we don't sequence the entire thing. What we do is we sequence off both ends, and then we read into the 581 base pair amplicon, and we assemble using the overlap regions, because again, we already know which lineages we're trying to discriminate between because of the data we have from the 580 base pair amplicon. And this just shows the sort of resolution gain that you can pick up by using this 2-tiered approach. Here's an individual where we were able to see quite confidently that the person was B07, B54. But once we added in the 1 kb amplicon, we were able to say that the person is B07, O201, and B54, B5401. And the same thing is true at the CELO side. And this approach has also allowed us to see limitations in the existing referenced databases for HLA class 1 alleles. Here's an example where we were actually able to identify that a patient was B15010101, B440301, C06, but that in fact they have a unique polymorphism in their C16 allele, which is one that had not been previously reported in the literature. So we believe that once we have this sort of full-length transcript profiling, we'll be able to create a much better database of HLA alleles, and we'll be able to rapidly accelerate HLA allele discovery. So to summarize this section, this sort of cDNA sequence-based typing is potentially a disruptive and cost-effective way of obtaining HLA data from HIV research cohorts. We capture more data at higher resolution than previous generation methods. It can be much, much cheaper, which allows us to contemplate doing cohorts that are hundreds or thousands of patients large. And we can extend known sequences in reference databases. And we're currently rolling this out, and our first widespread test of this is to determine the HLA types of HIV-positive patients who are long-term non-progressors in the Rakai Health Sciences program cohort in Rakai, Uganda. So now I'm going to move on to a project that we've been doing in collaboration with many of you here in Sao Paulo, where we've been using this technology instead of determining lots of HLA genotypes at one time, to instead look at lots of HIV drug resistance genotypes at one time. So the same idea, except instead of looking at HLA, here we're going to look at the virus and look at HIV drug resistance. So as many of you know, there's been an enormous global scale-up of antiretroviral treatment programs in the developing world over the last 5 to 10 years. But these programs have generally not been accompanied by a scale-up in laboratory monitoring to look for resistance, either in individuals or in communities. As some of the data that's been presented here has already said, and you may hear more in the course of the class, resistance to antiretrovirals can be a major complication in treatment. There's a large amount and potentially increasing amount of primary resistance, people who have transmitted resistant virus, and being able to understand how these factors are shaping HIV within communities is very important. But it hasn't been tested very much because, again, the per assay cost is very high. It's about $500 to $600 per test in the US. And it's required sophisticated Sanger sequencing instrumentation to do resistance testing. So we partnered with our friends from here, as well as our friends from Africa, Alesh Jani from Mozambique, Steve Reynolds, and Ron Gray from Uganda, and our clinical colleagues in Madison, including Jim Sossman, to see whether 454 sequencing could be used to look at drug resistance typing in different HIV clades. So we represent here largely clade C, clades A and D, clades B and F, and clades B. So we are looking, we're trying to get a pretty good snapshot at how well this technology can work across the entire global epidemic. And just in the last month or so, we've started collaborations with South Africans and Jamaicans who have contacted us because they heard we were trying to do this, and they were very interested in trying to adapt this technology to their laboratory instruments. The approach will look very familiar to what I showed for the HLA. We take plasma from an HIV-infected patient. We generate, in this case, 3 different amplicons that are overlapping. 2 of these amplicons span the RT gene. 1 of them spans protease. We pool the amplicons together, and we sequence them. By adding unique molecular barcodes to the ends of the PCR amplicons that are generated from each patient, we can pool together samples from multiple patients in a single instrument run. Again, this allows us to reduce the cost of generating the sequence on a per patient basis. And currently, we've gotten about 50 patients on a single GS Junior instrument run with 3 amplicons. That means that we're pulling together 150 amplicons. Doing that and keeping the amounts of each amplicon constant is a bit of a challenge, but I think that's something that we'll be able to improve as we go forward. And here's the sort of representative data that we get, showing about 70,000 reads from a single run. You see each of the 3 amplicons here. You can see that we didn't actually get proportional representation of each. We got much more of this amplicon, but we did get distinct peaks corresponding to each of the 3 amplicons that are in the pool. We've developed slightly different methodologies for studying samples with high viral loads and low viral loads. The important thing isn't how these methods are different. The important thing to remember is that you have to be very careful when you're looking at template sampling. If you have a sample that only has 100 copies of virus per milliliter of blood plasma, you can't generate 10,000 sequences or 1,000 sequences from that and expect to have those sequences be biologically authentic. There simply isn't that much template in the tube. And so we have to be very careful when designing these experiments to make sure that we don't try to overstate or oversell the conclusions. And this becomes very important when you try to use this technology to look for low-level variants. When you look for things that are present at 1% or 5% of the population, you have to be very careful that you're not simply resampling the same template multiple times. Here is some representative data from Brazilian patients. This was collected with Esther and Sabri and Esper and other people's help. It took us a while to get the regulatory approval into place and to get the samples to Wisconsin, but we were really heartened that when we looked in the first set of patients that we received, we found several examples where there were high-frequency variants identified in these acute samples that could correspond to transmitted primary resistance. Though it's important to mention that this K103N mutation, which we see 3 times, may be spurious because this mutation has not necessarily been associated with, is not always associated with phenotypic drug resistance. But nonetheless, it goes to show that with this sort of technology, you can, in fact, identify resistance mutations with a high degree of confidence. We also have 2 examples where we had paired samples from early, before pre-treatment and post-treatment, and we have mutations that increased in frequency from, in this case, 18 to 100%, and from here from 54 to 100%. So you can, in fact, use this technology to longitudinally monitor the emergence of drug resistance during infection. So currently, we're continuing to validate this method with samples that have been genotyped by other current generation technologies. We're validating it against different subtypes, as I mentioned. We're trying to establish the maximum multiplexing capability so that we can see how deeply we need to go. If we're simply interested in knowing whether a patient has some resistant virus or not, we can probably use a much lower depth of coverage than would be necessary if we were trying to identify minor variants that are present at a 5 to 10% proportion in the overall population. So designing experiments and multiplexing intelligently is very important. We're trying to improve our ability to do sequencing off low viral load samples. And then because it's often difficult to maintain cold chain on plasma and on blood, we're working with Tanuri and Rodrigo Brindiero and others to see about using this technique on dried blood spots instead of whole blood or flash frozen plasma. And so finally, I'm going to talk about how we've been using this technology in the lab to try to understand T-cell responses to HIV and SIV. Some of you who have been in the class in the past may recall that in past years I've talked about the use of the SIV-infected rhesus macaque and Mauritian cynomolgus macaque to try to understand aspects of HIV T-cell immunity that are really hard to study in people. And the reasons that we can use this animal model are that we can infect animals with the same virus. We can infect 8, 10, 12 animals with the same virus. We can not only infect them with the same virus, but we know exactly when they were infected, which is some of the data that I'm sure Ashley showed yesterday, showing that by knowing exactly when an animal was infected, you can precisely time the events of early infection. And this is something that we've been taking advantage of in trying to understand T-cell responses against HIV through the use of this animal model. And so we started with a very simple question. We know from work that's gone on over the last 15 years, many people in the audience here have been involved in this work, that T-cell selection and T-cell escape is a hallmark of HIV and SIV infection. Variability arises in the HIV genome due to T-cell escape. Several years ago, Todd Allen's group at Harvard tried to estimate how much variability in the genome was due to T cell escape. And we had done a similar study in SIV. And we both came to the conclusion that about 50 to 70% of the variation that arises outside of the viral envelope during chronic infection is driven by T cell selective pressure. But we've never been able to ask the converse question, which is how many different responses select for viral escape variants. So if you have 20 sites in the viral genome that are accumulating escape variants, so we know that a lot of the variation that arises during an infection is due to T cell selection. But we don't know if we see 20 sites in the genome that are accumulating variation, whether that means that there are 25 T cell responses that have been mounted in that individual, or whether there's been 75 T cell responses in that individual. So what fraction of all the T cell responses that emerge during an infection select for immune escape variants? And so to address this question, we took advantage of the same three MHC identical macaques that Shelby described earlier. So these animals have the exact same MHC class 1 and class 2 genes. They've all been infected with the same strain of virus, SIV MAC239. And for the purposes of this analysis, I'm going to exclude that fourth animal that controlled its SIV because it did have, as Shelby said, a different pattern of immune selection. We then waited about a year, and we sequenced the virus in these animals at 48 weeks post-infection by both Sanger and 454. So again, you saw this slide earlier. These are the three animals that I'm going to discuss. And again, the color of the vests refers to these animals' MHC types. The important thing to remember is that these animals are MHC homozygous and MHC identical to one another. So we would expect that they're going to mount the same CD8 T cell responses, or at least they would have the capacity to do so. So to ask the question on a genome scale, how many responses we're selecting for escape variants, we developed a strategy whereby we PCR amplified the entire coding genome of SIV in 3 fragments. We then use a process called tagmentation to introduce transposons into each of these amplicons to randomly shear them into smaller pieces that are about 300 to 500 base pairs long. We then sequence these fragments that have, we sequence this fragment library that's been subject to this transposition, transposon-mediated tagmentation. We generate hundreds of sequences at each site in the viral genome, so we get hundreds of sequences at each site through this entire 10 kb region. And compared to Sanger sequencing whole genomes, this has actually been both cheap and fast. It's very easy to generate these 3 PCR products, to randomly fragment them, and to sequence them. Here again is a schematic of the SIV genome. And because of work that Shelby had done with these animals that are MHC identical previously, we knew that there are 12 CD8 T cell epitopes, which are highlighted in blue, that these animals would likely mount immune responses against. Because the animals are MHC identical, we were able to ask the question, in each of these 12 epitopes, in each of the 3 animals, how many selected for escape variants? So we were looking at a denominator here of 36 epitopes. One of the first things that we saw was that when we looked in some of the epitopes that had been characterized as wild type by consensus Sanger sequencing, we discovered that there was actually an enormous amount of variation that was only resolvable using clonal 454 sequencing. The wild type virus is present at about 20 to 25% of all the sequence, but you see that there is this spectrum of viral mutations within this epitope that we completely missed by consensus Sanger sequencing. Perhaps not surprising, but nonetheless affirming some of the potential of this technique. And so when we summarized over the 36 epitopes, again, 12 epitopes per animal, 3 animals, what we found was that there were 10 epitopes that were wild type by both Sanger and pyrosequencing. There were 14 epitopes that we had identified previously as wild type by consensus Sanger sequencing, but were actually variable by pyrosequencing. There were an additional 11 epitopes where we had identified variation by Sanger sequencing, but detected more extensive variation by pyrosequencing. And there was only one epitope where the escape mutation in the epitope was essentially fixed, and we detected the same amount of variation by both Sanger and pyrosequencing. So in sum, 70% of the epitopes that we believe to be recognized in these animals selected for escape variants by approximately 1 year post-infection. And so just to summarize, these mutations were often seen in multiple animals, the same mutations, which suggests that these are real escape mutations and not spurious artifacts. Because we have the ability to see which amino acid changes are linked within the same sequencing read, we're able to detect changes that affect 2 or more positions within the epitope, which is something that happens more commonly than we previously appreciated. And finally, and I think this is going to be the provocative point that I'm going to spend the last few minutes on, I think that with this new technology, we can really start to use variation that's detected with these methods as a proxy for understanding the immune responses against these viruses. And I'm going to share a little bit of very recent data that's still being analyzed with you in hopes of convincing you that this is the case.